Right.
So, welcome everyone to this EAAFP Shorebird Working Group working webinar on monitoring shorebird habitat quality. As I'm sure almost all of you know, the East Asian Australasian Flyway Partnership is an informal voluntary international partnership launched in 2006 to conserve migratory waterbirds and their habitats along the Flyway. The goal of its Shorebird Working Group is to promote, facilitate, and coordinate shorebird conservation, management, education, and research activities within the Flyway, and to improve communication between shorebird specialists and managers. The working group has about 80 members, including EAAFP partner reps and invited experts. The Shorebird Working Group's 2026 to 2030 work plan centres on 4 themes: monitoring, conservation of species and habitats, capacity building to promote science for conservation and management, and communication. The webinar has been organised by the Shorebird Working Group, and I think it's going to touch on a number of those 4 themes in the work plan. We're also very thankful to the EAAFP Secretariat for their support in implementing the webinar, providing the Zoom structure, etc. For those of you I don't know, my name is Misha Jackson and I'll be chairing today's webinar. I'm a member of the core team of the Shorebird Working Group, and I'm also a research scientist at the Commonwealth Scientific and Industrial Research Organisation, but that's very long, so most of us who work here call it CSIRO. CSIRO is Australia's national science agency and one of the world's largest multidisciplinary research organisations. So I'm based in Canberra in Australia. I did my PhD about managing artificial coastal habitats to benefit shorebirds. I've done some shorebird catching and tracking. I've worked at some NGOs and universities. In my current role at CSIRO, I mostly work on waterbird tracking in the context of inland water and wetland management. So that's more than enough about me. Let's get going on the webinar itself. The rationale for having this webinar is that to help understand population trends that we see in the shorebirds of the Flyway, and also to help with their conservation and management, it's really critical to measure habitat quality. But this can be really challenging to do in practice. There's lots of elements of habitat quality that affect especially the long distance migrants. And you'll see from the talks today that there's many different things to think about when we say habitat quality. And so the aim of this webinar is just to bring together researchers and all of you interested in shorebirds throughout the Flyway. The webinar speakers will be pretty brief and present some new science on measuring all different aspects of shorebird habitat quality. And we're kind of moving north to south along the Flyway as we talk about these different elements of it. And after the talks, we should have significant time left for discussion. And the idea is that will allow all of you to share your thoughts and experiences on this topic and help us consider together how additional habitat quality monitoring might be implemented in different parts of the Flyway. And I'll try and structure our discussion so that we sort of stay on track and hopefully tackle a couple of key questions. So without further ado, we will move to our first speaker. Julia, do you want to just share your screen? I'm going to introduce you, but maybe just make sure that the screen sharing is working.
Yeah, I'm having technical difficulties, sorry everyone. So would it be possible for me to go second? I'm trying to download my slides.
Happy to go first. Okay, no worries. We'll give you a few minutes to get sorted. In that case, Tong, if you're happy to share your screen, that would be wonderful. I'm sure many of you already know Tong Mu, but Tong is an associate research scholar at Princeton University, broadly interested in the ecology and conservation of migratory birds. His research focuses primarily on studying and protecting migratory shorebirds and their coastal habitats in the EAAF. Combining field survey, individual tracking, mathematical modelling, and data mining, Tong's research aims at delivering feasible and effective conservation insights through identifying vulnerable species and key habitats from multiple angles and across different scales. In addition to his research, he's also really active in raising awareness and promoting biodiversity conservation through science communication, stakeholder engagement, and policy advisory. So I'm sure many of you know Tong, really looking forward to his presentation, and looks like all good with the screen sharing. So over to you, Tong.
Perfect, thanks. Thank you, Misha. You hear me okay? Yeah, well, thanks for holding and hosting this webinar. The title of my talk today is Quantifying Habitat Quality is Key to Conservation Success. It's a summary of some insights from a collection of some new studies of my collaborator and I that's published recently. We'll focus mostly on stopover site, but towards the end, during the discussion, we'll also talk about how this may be applied to non-breeding habitat during the wintering period. So let me begin the talk by highlighting some of the unique challenges in studying and conserving migratory species, using spoonbill sandpiper, which is a flagship species along the flyway as an example. They depend on many sites every year to survive, to migrate, and to breed. And across this site, they face many different threats, including coastal development, hunting bycatch, invasive species, sea level rise, etc. And the spoonbill sandpiper has been estimated to be declining at a rate about 26% per year. Luckily, with a dedicated global effort, including very intensive monitoring along the flyway, head starting, habitat protection, tackling illegal hunting, spontaneous removal, people have been able to slow down the population decline by fivefold. Now it's estimating to declining at a rate at just 5% per year. So from an optimistic point of view, it's a great conservation success. But this 5% per year is still much faster than most of the EAF shorebirds declining. And it also means that there will be possibly less than 100 individuals left in just 30 years. And even more worrying is the fact that we seem to have exhausted our arsenal with this very comprehensive list of conservation actions. The birds are still declining. What else can we do that we have not done yet to save the spoonbill sandpiper or to slow down their population decline? So the two key questions in my mind in conserving migratory shorebirds are, given the different threats we know that they're facing, how can we quantify the contribution of those different major threats to their population declines, and how do we prioritize our conservation actions? And second, perhaps even more challenging, important, but often neglected, is how can we evaluate the conservation effectiveness or success? How do we know whether we're doing good enough? So this is where I think monitoring habitat quality can really help. But an important fact that is possibly there in everyone's mind, but often overlooked, is the fact that population change and habitat change are often decoupled. What I mean by that is that imagine we have a very simple migration route of the same of a population, migratory population. We have one breeding site, two stopover site, and a non-breeding site. So the outline of the circle here means habitat quality, how many birds it can support, whereas the fill means how many birds it actually has at the moment. So imagine that all the sites started being able to support 100 birds, and they all support 100 birds. So now, if stopover site 1 suffers some habitat change, it can only support 50 individuals. We can imagine that the number of birds that passing through that area is also 50. But because it's the same population going through all those sites, even though there's no habitat change at other site, they can also only support 50 individuals, which means that if we detect local population changes, it does not necessarily mean local habitat has changed. And now imagine stopover site 2 also suffers some change, but because it has also suffered some changes and it can only support 50 individuals, but because it only supports 50 individuals at the moment, so we would not detect any population changes at this site and also at other site, which means that sometimes local habitat changes does not necessarily means the local population will be affected, nor the global population. So monitoring shorebird is very important, but not enough. It cannot tell us how good habitat quality is and how habitat change may affect migratory populations. So what we should do is that we simply need to move beyond just monitoring birds as an index of habitat changes, but we need to monitor bird and habitat directly and simultaneously. So in terms of birds, we also need to improve the metrics we're using. what has been commonly used in monitoring birds is the peak or the total abundance, which is also the widely used criteria for recognizing Ramsar site or important bird areas. However, at stopover site, the population of migratory species is always changing, shown by this solid and dashed line here as two examples of stopover site. If we base solely on the peak abundance, which is the maximum amounts that we see on a given day, we may conclude that this site shown by this dashed line is more important than the solid line site. So we would argue that both the abundance and the duration of habitat use affect how shorebirds use the site, particularly how much resources they need from those stopover habitat. So we proposed to quantify habitat use intensity as bird days as a better measurement of the bird's demand on the habitat, and which is effectively the size of the area under this abundance curve. It takes into consideration both the abundance of the bird and the duration of their habitat use, and it can be derived very easily from existing monitoring data without much additional effort. So similarly, to be able to directly compare the demand and supply, we also propose to monitor and measure the carrying capacity from food availability in the habitat and the intake rate of migratory birds, resulting in a supply measurement of the habitat or the habitat quality measurement also measured in bird days, so the demand and supply can be compared directly. So this demand and supply framework has recently been published with more detailed account and explanations of the rationale behind it and the strengths of the framework. We also have a case study demonstrating how it has been applied with more details on the methodology in doing that, focusing on migratory shorebirds and habitat monitoring. So some of the key insights we gained from those two studies. First, that monitoring bird alone is not enough because of this decoupling of habitat quality change and population changes. And second, that we need to measure the demand and the supply both in bird days or similar metrics that takes into account the temporal changes in those two aspects. And based on those two metrics, the important sites where many birds concentrated that we normally identify may actually be used well below its capacity, which means that the site can actually support more birds in it currently support. So habitat loss may not be an imminent threat to those important sites, however many individuals it's supporting. And the site where conservation or restoration effort is most urgently needed is not where the birds achieve high habitat use intensity, but where this use intensity is close to its carrying capacity, because any changes to those sites, whether it's a decrease or increase in the carrying capacity, will likely affect both the local and global populations. So I think in addition to help identifying conservation priority sites, habitat monitoring can also help us identify the priority areas within each site. Simply because we need to do very comprehensive monitoring of habitat quality. We were able to document where the foraging area of the red knot is at the site, which is shown in this area. And we were also able to confirm that this distribution is driven mostly by the distribution of their food resources, the tiny bivalves, shown by this colored gradient, showing where the bivalves are. We have done some similar studies across different shorebird species, across different sites along China's coast, asking where do shorebirds concentrate on the tidal flat, and where does their food concentrate, and also where do the most prominent threats come from on those habitat. And the key message is the same, is that the tidal flat actually highly heterogeneous. The distribution of the bird, the food resources, other environmental factors, and the threats, all of which are not uniform across the same tidal flat, which means that if we look at habitat loss just as a percentage, it does not represent the percent of how much habitat quality has lost. It does not mean how much population will be affected. And in fact, some small loss of habitat can lead to large declines in the population or the habitat quality at stopover site. as Mitra mentioned, measuring habitat quality is not an easy task, especially for migratory species. There's a lot we still don't know about the diet, how the composition, the flexibility, the seasonal or site variations, and the diet of competitors of the shorebirds. And direct measurement of their food quality is sometimes very labor and time intensive, and the sampling design will vary with global or local conditions. So there were some more recent development in using other environmental metrics or indirect quality metrics, which you will hear shortly from Mitra. But also there's a study coming up from Siam, who's having a study combining satellite tracking with remote sensing data to evaluate habitat quality. And you'll hear Mitra from Mitra soon about her study in Australia using indirect measurements of habitat quality. But I hope I was able to convey the message that marine habitat quality is challenging, but is key to evaluate or to ensure conservation success. And if proper sampling of habitat quality of more than 30 sites for more than 10 years can be done by a dedicated team of people, I feel that quantifying habitat quality is not only something that's something that we should do, but it can also be done. Thank you. That's all I have. Happy to take any questions.
Thank you so much, Tong. And if everyone throughout this webinar, both after the speakers and during the discussion, if people could just raise their hands if they want to ask a question or make a comment. I think it's too hard to go back and forth with the chat. So do we have any immediate questions for Tong? You can also have a think about it, and we'll definitely discuss some of these aspects at the end as well. Any immediate questions?
Phil has his hands raised.
Oh, we can't hear you. The old mute.
There we go. Yes, the importance of monitoring, and but one of my the questions is just the frequency of monitoring, not just peak numbers. Peak numbers could be birds just resting there, and the habitat may not be sufficient to maintain them. So I'm a great believer in doing monthly monitoring at all my sites, but that's a big strain in some, according to some people in Australia, to get people to do things more frequently. But again, if you only have one or two counts or two counts a year and you have a bad weather, then you've already got records of one. So I think that the frequency of counting is very important. All of your other points are valid as far as I can see. It's a very good presentation. Thank you, Tong.
Thank you, Phil. Maybe just to phrase that a bit as a question for you, Tong. I mean, in that stopover site context, in order to do the types of measurements you're talking about, how frequently do you feel that the sites do need to be monitored to be able to, you know, have a good estimation of that area rather than just the peak?
Yeah, that's a great question. And so I would think the most optimal frequency may depend on the species composition and the specific patterns of arrival and departure at those sites. But the currently what has been done along most of the China's coast at least is that people are doing bi-weekly counts, and that's a good enough frequency to estimate that area under the curve. And I think the point I want to make is that we can actually have better usage of or better use of the existing data by looking at the area instead of trying to pick out which one is actually the maximum. By drawing that shape and calculating the area, we actually provide us with much more information about the bird dynamics and the intensity of or the importance of those sites. Because as Phil mentioned that the peak account depends on the weather, on the frequency you're doing the site and the species. So it's very sensitive to the to the how to say the condition, the server condition that you're there. whereas the shape is less sensitive to the actual count to each date that you're doing the monitoring.
Great, thanks, Tom. I don't see any other hands right now. One other one. We'll take one more question and then on to the next talk. Maxime, have you got your hand up?
Yes. Thank you, Tong, for your presentation. That's great. I was wondering, how did you estimate the carrying capacity in such a continuous metric? Thank you.
Yeah, so we were lucky in a kind of a special system where the amount of food doesn't change that much during the period when the birds are actually stop over at the site. So it's a fixed amount of the resources. So I think for more dynamic one, you have to account for the growth and the increase in the food resources. For example, if we're doing uh, current capacity estimate for migratory land birds, when the amount of worms or amount of fruits are constantly being produced, then we'll have to account for the increase, the rate of replenishment. But for most of the shorebird site, my hunch is that within the period the birds are visiting, most of the well, if it's a benthic, if it's not something that's uh, in the water, if it's benthic invertebrates, then the amount is more or less fixed during the period when they're visiting at stopover site at least. It's going to be quite different if it's in Australia or in New Zealand or in Southeast Asia, where they're there for a much longer time of half a year. I don't think that can be assumed. So we'll have to understand the food dynamics more as well. That's definitely a huge gap.
Yeah, okay. And do you use giving up rate as an indirect
Yes.
measurement? Yeah.
Yes, we did. We estimated the give up density. Uh, that's what you meant, right?
Yeah, sorry.
Yeah,
Yeah.
we estimate the give up density in the field by looking at the distribution of the birds, where they're not foraging, where there's still food but not foraging. We set a threshold as the give up density.
Okay. Thank you, Tong.
Yeah, thank you.
So our next speaker is Yanju Ma, and unfortunately Yanju couldn't join us because of the awful time difference between here and Europe, and we couldn't get a time that wasn't at 2 a.m. for somebody. So we have a wonderful pre-recorded talk. But for those of you who haven't met Yanju, Yanju Ma is an ecotoxicologist with a great interest in understanding how environmental pollutants shape ecosystem health. Most particularly, she's interested in mercury, the global concern, in threatened animal groups across multiple ecosystems. So she's looking at langurs in limestone forests, shorebirds in our flyway, and wildlife inhabiting the Himalayan region. So really interesting stuff. Her research aims to advance conservation by improving our understanding of pollutant threat and its impacts on wildlife populations. So I am hopefully going to share my screen with a video this time. We tested it and it worked earlier, so fingers crossed everyone that this will work. So I need to say the sound thing, don't I?
Yeah, and it's share sound. Yep.
Okay.
Yep.
Let's see how this goes. Full screen and play.
Hello everyone, I'm so happy to be here to share some work we have been done in mercury with some migratory shorebirds along this Friday. The topic is mercury houses to migratory shorebirds along the EAF and identify hotspots and species at risk. I'm Yanji. Sorry I won't be able to join you online, but if you have further questions, please don't hesitate to reach me through the email address. When we talk about habitat quality, there is threat pollution. Actually, it can degrade the habitat qualities in the areas such as the coastal area and restaurants, where the shorebirds heavily depends on. Mercury actually is a global concern. It has been proved have many negative effects on birds, particularly for migratory species. It can, for example, it can rapidly accumulate high levels in organs, extend motor period, impair takeoff, alter stopover decision, reduce flight performance, and even decrease survival. Asia actually contributes half of the global mercury emission. Mercury, like birds, can migrate long distance and reach major breeding grounds of migratory birds, such as Arctic. Wetlands are significant areas for mercury maceration, which leads to its bioaccumulation and biomagnifications along the food chains. So when we think about mercury levels and this potential hazard to migratory shorebirds, we should be able to understand some questions such as what's the levels along this flyway, any hotspots and groups at risk, and the factors affect mercury levels. That will be crucial for shorebirds conservation. Feather mercury actually indicates the mercury burdens at the molting site. For example, juveniles actually they mold their fly feathers at breeding grounds, which have information about the breeding grounds for contamination. Adults, they generally replace their fly feathers at stopovers or wintering site, so their feather mercury actually indicates their body levels at these sites. From our previous work, we found that primary cover 6 actually nicely capture the median mercury levels along the mold sequences. So we collect feathers from 18 sites of China's and Australia sites, and we have some we measure mercury levels and also use the race assessments from the previous studies. When we look into the results, we have about 1,000 individuals belonging to 33 species. We can see that nearly 1/3 of the samples is the toxicity benchmarks, and 5 were at moderate rates, and 1% were at high and severe rates respectively. So we look into species variation. We can see that marsh sandpipers and common lichens actually have very high rates, high percentage at risk. These two species actually belong to Changa genus, and with a preference of inland wetlands and agricultural habitats with vegetation cover, which makes sense because maceration generally happens to these habitats. And we also look into the factors. We can see that through the linear mix model, we can see molting regions is spread the most, and then the habitat preference and diet and the foraging behavior also contribute, but less pronounced. We look into different life stage. We can see that breeding grounds was non-breeding ground. Non-breeding ground have much higher mercury level, about 1.5 fold. We can see the South China, such as Hong Kong and Zhejiang Peninsula, they have much higher mercury level comparing to Yellow Sea. Actually, Yellow Sea have similar to Australia, comparing to other breeding grounds like temperate zones and Arctic region. And we have a student Wu Yin also continue working on some factors collected in Southeast Asia, and we have more large sample size now. When we look into South China and Yellow Sea and Southeast Asia, we found there is no difference among the three regions, which indicate that South Asia also can have some hotspots there. Take home message: we cover most of the important sites along this flyway. This is a flyway scale mercury pollution study. We found the non-breeding grounds such as Southern China and Southeast Asia with good sample size have higher mercury level. Species such as Changaginians, like common lichens and marsh lichens, have many more potential. In the future, we can combine with tracking data to improve our understanding, because with tracking data, we probably can have more information about their habitat use, which is crucial for understanding why some species particularly at risk. With that, I want to thank our volunteers, especially the Hong Kong water birds training group, and David, Simbad, and Toby, our founding source in Shenzhen's Science and Technology and Guangxi Normal University, and a bunch of the other founding resources. For more details, you can visit the research already published in Communication Biology 2024. If you have further questions, you can reach me through the email address below. Thank you very much for your attention.
Okay, so obviously we can't have any live questions, but I was a little bit cheeky. I got the talk ahead of time, and so I emailed a question to Yanchu. So you'll have to bear with my question. My question was, do you think we should be monitoring mercury levels regularly at shorebird sites, and how would this kind of work? And Yandu's thoughts on this were as follows. She said, ideally, we should implement regular biomonitoring at key shorebird sites along the flyway. At minimum, monitoring environmental matrices, so things like sediments and soils, would be valuable. But interpretation can be quite complicated by site-specific factors such as organic matter content and other geochemical conditions. And so, where feasible, incorporating biological indicators would strengthen the assessment. Non-invasive samples like feathers, as well as minimally invasive samples like blood, can provide insight into mercury exposure in birds. In addition, monitoring key prey items, such as polychaetes and bivalves, would help link environmental contamination to dietary exposure pathways. Overall, a combined approach integrating environmental and biological monitoring across representative sites would likely be the most effective strategy. That makes a lot of sense to me. And I was thinking that, the diet studies are always really time intensive, and so maybe there's an opportunity, if we're doing other studies on macroinvertebrates, that this could be something that also gets incorporated into that. So yeah, very interesting. So I won't ask if there's other questions because none of us will be able to answer them. But as Yanju said, please do email her if you have specific questions, and maybe this pollutant aspect will also come up during the discussion. I know some of the co-authors as well on some of that work are here. So we do have some people who might be able to talk to some of this as well. All right, with that, I will share my screen, I hope.
I can ask people to put questions for you in the chat and you can pass them on.
Yeah. Is that working on your screen?
Yeah, just it's coming up. Yep, it's there now.
Okay, fantastic. All right, so just to round off the kind of talk portion, I just wanted to touch on a couple of other aspects of habitat quality, and then I'm really keen to hear what everyone has to think about this topic. As is customary in Australia, I'd like to begin by acknowledging the Ngunnawal people as the traditional owners of the lands that I'm joining the webinar from today, and pay my respect to their elders past and present. I just wanted to mention this study at the beginning because it is kind of it was sort of the foundational work that led to the field study I'm going to talk about next. If you're interested, you can use the QR code to follow it. I think it is a nice sort of summary of the global literature on how people have gone about tackling this question of habitat quality for water birds across the world and across time. So this was led by a colleague of mine, Rowan Mott. And one of the things I just wanted to point out, we've talked about this a little bit in other talks as well, is just to keep in mind that one of the really tricky things with habitat quality is that we're usually not measuring things directly. So it's really hard to measure, especially migratory shorebirds, to measure reproductive output and survival directly and think about these. So we're often measuring things indirectly, all of these habitat quality proxies, and then we have to figure out some way of guessing what the relationship between those and direct demographic variables are. So it is really challenging, and there's a nice discussion of that in this paper if you're interested in thinking about that a bit more. But the study I'm going to talk about took place in the Coorong. So for those not from Australia, the Murray-Darling Basin is Australia's largest river system. So on that red circle on the left map, that's showing the Coorong, and then that's a zoom in on the right. And you can see it's quite a unique wetland. It's a really long, really narrow wetland system, about 110 kilometres long, that's internationally important for water birds. And it's got this kind of pinch point in the middle. That's why we've got the blue and the red. So it's two lagoons. They are connected, but there's a really narrow pinch point between the two lagoons. And so it doesn't actually connect with the coast directly, except at the very top. So at the northern end, it's connected to the ocean through the mouth of the River Murray, which is at the very, very end of that massive Murray-Darling Basin. And so we have an interesting salinity gradient that goes down along the wetland. So a lot of lessons from the Coorong are not easy to transfer to other sites because it's such a unique configuration and quite unusual. But there's a couple of things from this study I just wanted to mention that I think are kind of widely applicable. So the shorebirds we looked at in this study were 4 migratory shorebirds, mostly the small ones, sandpiper, curlews, sharpies and curlew sands. And then we also looked at some non-migratory shorebirds, although within the continent, redneck avocet is actually moves quite a lot. But local birds, there's lots of little red-capped plovers. So this is mostly a study on smaller shorebirds. Okay, so we know that abundance is a commonly used proxy for waterbird habitat quality. So we did look at abundance and we said, okay, we've got this opportunity with this nice long narrow wetland system to go, why are the birds distributing themselves along this long narrow space? What is it that attracts them to some areas and not to others? And so we had these samples that went all the way down the wetland, sort of 3 in the top, 3 in the bottom, and 1 extra on the side. And we just did 2 on each sampling trip, which we did throughout a whole year. We did 2 20-minute counts, 1 in the morning, 1 in the afternoon. But we also tried, and I think this actually did work really well. And this is something I wonder about just supplementing regular counts with this, is we did use some camera traps. So we put out these camera traps for 6 to 8 days per visit. And you might think, oh, it would take so long to go through all those photos. But we actually programmed it just to take a photo once an hour at the waterline all day and night. And so that's not actually that many photos to go through. And so I think this is something that could be looked at other sites as well, just to see that, more continual indication of abundance. Okay, so then we thought about, what might influence shorebird abundance, and these are the things that we measured that we could sort of think of and feasibly measure. So we looked at time of day, exposed mudflat area, shallow water area, salinity, what the wind was doing, that's kind of particular to that wetland, it's really important, temperature, and then we also looked at preferred macroinvertebrate prey, and that was based on some diet studies that had been done recently in the wetland as well. And we just measured, as I'm sure a lot of you have been involved with studies, just doing up to 3 centimeters depth using one of these core samplers, and we just measured at the waterline. It's very time intensive to both do the sampling and to go through the stuff. It really is quite a big job. Okay, so what did we find? We did a bunch of linear mixed models to say, okay, which of these things is important? So what we found was that the most important indicator of shorebird abundance was the area of exposed mudflat and shallow water put together. Time of day also did have an effect, as you'd sort of expect. But the interesting thing was we found no relationship with the macroinvertebrate prey really at all and shorebird abundance. And so I think the interesting lesson from that, and something to think about, is that it sort of suggests, and we actually do think this might happen in the Coorong, that shorebirds are susceptible to ecological traps. Apologies to anyone that's not a Star Wars fan. I just couldn't think of any other way to express it's a trap. But yes, so what could be happening is that if we have big areas of habitat, particularly if we have any control over the availability of shallow water and mudflats, if there's not actually good food resource in there, we could be pulling shorebirds into areas that actually are not high quality. And we might think that they're high quality because we can see a lot of birds there. So it's important to kind of look a little bit deeper. And so the kind of follow on from that for us was we had a go at observing some shorebird feeding behavior by taking lots of videos like this. And we did some more modeling, looking both at the pecks and probes that the birds were doing, but also the steps that they were taking. And the interesting thing we kind of found from this exercise was that, especially I think with these smaller shorebirds, it's really difficult and time consuming to measure pecks and probes because the bird's behavior can be a bit hard to interpret. But also we didn't really find any relationship between those measured pecks and probes and the macroinvertebrates. And we think that this could be that in small shorebirds, it's really hard to tell if their peck was actually successful. So they might be pecking at the substrate quite a lot, but not actually finding food to eat. And it's quite hard and then even more time consuming to try and assess if it's successful. On the other hand, we found that their step rate was actually really quite easy to measure. And I think now you could really easily get an AI to do it with the videos, just to count the steps within a period of time. And we found a really nice relationship showing that, basically birds took way less steps if there was more food. If there was no food there, they were running around everywhere looking for food, which does make sense. And so if you wanted to keep track of this food resource aspect of habitat quality over time, perhaps measuring step rate could be a good way. It's way less time consuming, less invasive, less disruptive, much easier than obviously taking millions of those core samples. And it's still taking a cue from the birds about what's going on, but it's easier to interpret. And we found a much stronger relationship than worrying about the actual intake rate of the birds. So that's something from this study I thought might be interesting. So that's all I wanted to say about that study. If you're interested in the details, you can follow the link and read the whole paper. But just before we finish the kind of formal talk aspect of this webinar, I did think it would be remiss not to mention one other aspect of shorebird habitat quality that those of you who know me know that this is a passionate area of study for me. So in coastal habitats, as we know, there's really two normally critical habitats for many species. So we've got the tidal flat feeding grounds, and then we've got high tide roosts. And if we look at satellite tracking data, we often see patterns like this. So birds are spread out on the tidal flats and they're going back and forth to the same roost sites. Okay. The challenge with this, of course, is that those high tide sites are often situated in really high use areas of the coast. Lots of people, lots of activities, in some places, lots of production. And so as a result, birds often use artificial habitats, and these can be private or industrial land. Paper we did a few years ago, basically just looking across the whole flyway, is showing that throughout all the flyway, shorebirds use lots of different artificial habitats at high tide, but everywhere they use artificial habitats. Okay. Again, I won't dwell on this too much because a lot of you have seen this before, but I just wanted to mention these high tide habitat management guidelines that we made in 2021. To put these together, we had a workshop, we looked through conservation evidence documents, and we had fantastic written input from 19 different experts around the Flyway. Some of them are here in this webinar today. That was fantastic. And then myself and Phil Straw, who you heard from earlier from AWSG, sort of edited them together. So the content of these guidelines was just to say, firstly, how do shorebirds choose where to roost, and how can we explain this in a simple way to non-shorebird experts? And so we talk about things like avoiding predators, avoiding disturbance, providing feeding opportunities even when tidal flats are covered, and minimizing energy costs. Okay. Then we said, well, what are important roost site features? This is a really well-used roost site in Darwin. It's within the port. Why do the birds go here at high tide and not go somewhere else? Why is this a preferred roost site? And we talk about things like water cover and depth, vegetation, sight lines, prey salinity, temperature, distance from feeding grounds. Okay. And then we talk about some basic principles for managing roost sites. So things like limiting disturbance, managing visitors, engaging with the community, both about this disturbance aspect, but also about why roosts need to be the way they are. When you look at a shorebird roost that's really good, imagine that picture from the previous slide. It doesn't look that great to someone who doesn't know shorebirds well. You think, well, it'd be better if there was some nice plants here, and it might be better if there was more water, but shorebirds like these really particular features. And so explaining to the community why the features are important for shorebirds specifically is really important. And then, of course, you've got to do your vegetation and water management to maintain those features. So, and I also did want to mention that we had some amazing support from AWSG to translate these guidelines into a number of Flyway languages. My point of bringing these up again, I think, is that in the sort of in contrast to some of the other talks we've heard today, where on tidal flats and up on the breeding grounds, there's a lot of aspects of measuring shorebird habitat quality that are still have some unknowns and can be quite challenging. I think with high tide habitats, we kind of have a pretty good idea of which aspects make for high quality roosting sites and why, and even how to manage them. But I wanted to bring them up in this webinar because something I wonder about is monitoring this. So we know that people do amazing shorebird counts, sometimes very regularly all across the Flyway, but personally, I only know of a few examples of where people are trying to monitor these aspects of high tide roost habitat quality. And I wonder about things like, how do we make this happen? What would we measure if we wanted to have a robust measurement of this over time? And also thinking about, at what point, if we think conditions are deteriorating at a roost site, the quality is going down, what's the point in time that we need to have an intervention to maintain the higher quality aspects of those roosts? And I know there's quite a few people on the call who are interested in this topic and have some experience in this. So I just wanted to raise it because I didn't want to leave the high tide stuff out. And I think it's also potentially an interesting thing for us to discuss together.